السلام عليكم. محاضرتنا لهذا اليوم هي عن الباثوجينيك intestinal protozoa especially الانتاميبا هيستوليتيكا by دكتور رشا assistant بروف جامعة بابل كلية الصيدلة فرع العلوم المختبرية السريرية. المحاضرة هي المرحلة الثانية كلية الصيدلة. بالبداية ناخذ فكرة شنو البروتوزوا. So protozoa are macroscopic one-celled organisms free living or parasitic in nature able to multiply in a human. يعني انه البروتوزوا هي عبارة عن طفيليات تتكون جسمها من خلية واحدة وتعيش يا اما حرة بالطبيعة او تكون متطفلة وتقدر تتضاعف جسم الانسان. Protozoa can be classified based on first mode of movement and second the main site of infection. So the classification based on the movement, protozoa can be classified to Sarcodina, the amoeba, example amoeba pseudopodia, وهنا ناخذ عليها مثال example الانتاميبا. Second السيليوفورا واللي هي الهدبيات the ciliate, example بلانتيديوم كولاي. Third الماستيجوفورا اللي هي الصوتيات او نسميها الفلاجيليت example جياريا ليشمانيا and trypanosoma. Four sporozoa the organisms whose the adult stage is not motile example plasmodium and cryptosporidium. Second classification based on the main site of infection. So according to the main site of infection, we can classify the protozoa to three species or three classes. First one, intestinal protozoa, example amoeba, giardia, and palletidium. Second, urogenital tract protozoa, example Trichomonas vaginalis, is the most important pathogens. Third class, the blood and tissue protozoa are varied group consisting of flagellate, trypanosoma and leishmania, and sporozoon, example plasmodium and toxoplasma. محاضرتنا بالذات اليوم هي عن الساركودينا او الاميبيك سبيشيز اللي هي تتضمن او تنتمي الى الانتستينال بروتوزوا. الاميبيك سبيشيز اللي هي اولا او first Entamoeba Histolytica, it's a pathogen. Second, Entamoeba hartmanni. Third, Entamoeba coli. Fourth, Entamoeba pollici. Five, Endolimax nani. All of these are non-pathogenic or cause asymptomatic infection, except Entamoeba Histolytica. Other species are Naegleria, Fowleri, and Entamoeba species. These species can cause disease but not in the intestine. Today we will talk especially about Entamoeba Histolytica, which is the pathogenic in the intestinal intestine. The habitat of Entamoeba Histolytica is a common parasite in the large intestine, reside in the mucosa and submucosa of a human, and in the certain other primates and some other animals. Entamoeba Histolytica derived its name from its ability to lyse virtually every tissue in the human body and in the bodies of the experimental animals. Many cases are asymptomatic except in human or among animals living under stress, example zoo, hound primates. Morphology of Entamoeba Histolytica. These parasites have two stages in their life cycle or two forms in their life cycle. These are trophozoid and cyst. Trophozoid is the active vegetative amoebas form, while cyst or intermediate pre-cyst and mature cyst, these are inactive stage, infective stage, and resistant. We will start with the first stage, the trophozoid or amoeboid trophozoid. Trophozoid, the only form that presented in the tissue causing disease, which means it's pathogenic stage. It's also found in fluid feces during amoebic dysentery. Its size is from 15 to 30 micrometer. The cytoplasm of trophozoid has two zones, a hyaline or clear outer margin ectoplasm and granular inner region endoplasm that may contain red cells, pathogenic features, leukocyte and tissue debris within the food vacuoles, but ordinarily contain no bacteria. The presence of ingested erythrocyte is the characteristic feature of Entamoeba Histolytica. Trophozoids are feeding, motile, with active unidirectional and purposeful motility. So its movement results from the presence of long finger-like pseudopodial extension of ectoplasm, filled with endoplasm. Let's move to the second form of Entamoeba Histolytica, which is cyst. Cyst, it is smaller in size than trophozoid, spherical, varying from 10 to 20 micrometer in diameter. Food vacuoles and chromatin bar disappear in the mature cyst. Present only in the lumen of the colon and in mushy or formed feces, but not in tissue. Surrounded by a thick wall, which make it highly resistant to the gastric acid, adverse environmental condition, and the chlorine concentration. The initial cyst contains from 1 to 2 nuclei, while the mature cyst contains 4 nuclei. Diagnosis in most cases rests on the characteristic of the cyst, since trophozoids usually appear only in direct feces in active cases, and also survive for only few hours outside the body. Stools may contain cysts with 1 to 4 nuclei, depending on their degree of maturation. Let's move to the life cycle of Entamoeba Histolytica. As I mentioned before, Entamoeba Histolytica life cycle have two forms in the life cycle. So the infection started with the ingestion of of cyst, the infective stage, with the contaminated food and water. The cyst, because it has a thick wall, they can resist the gastric acid. So, when they reach to the small intestine start to release the trypsin enzymes that lines the wall of the cyst. And the metacyst ampulla so escape from the cyst. This trophozoid or metacyst trophozoid divided by binary fission, each nucleus will give 2 nuclei. So the total, each cyst will release 4 trophozoids. Then the cytoplasm separates and release the trophozoid. With the fecal stream, the metacyst trophozoid arrive to the large intestine. They multiply and then they invade mucosa and lodged in the submucosa. In the submucosa, they grow and multiply, replicate, feeding, and then produce proteolytic enzymes that cause the destruction and erosion of the mucosa and submucosa in the large intestine, causing the flask-shaped ulcer. In the peripheral of the lesion, the trophozoid appear that excreted with the feces, with mucus and blood. At this stage called Dezentral. Sometimes the trophozoids cannot cause disease or cause a symptomatic infection. And in few cases, they can enter the radical ventral veins and arrive to the liver. So it and causing extraintestinal amoebiasis, which means liver abscess. So when the immunity of the person developed and the condition become unsatisfied to the trophozoid discharge the food vacuoles and chromatin bar and begin to transform to the cyst. At first, pre-cyst, then uninucleated cyst, then the nuclei divided, cause, and release. Binucleated cyst, finally transform to the tetranucleated cyst. This process, the transformation of a trophozoid to the cyst, called encystation, and this process happens only in the large intestine. So, the process at first, when the cyst transforms to the trophozoid, called excistation, and happen only in the small intestine. Finally, the cyst excrete or exist with the feces. Disease and clinical findings of Entamoeba histolytica. So Entamoeba histolytica can cause 3 forms of disease. Intestinal amoebiasis begin with the abdominal discomfort and episode of diarrhea or varying duration, at first mushy, then increasing mucus, including blood, tinge, so-called red currant jelly stool or dysentry. Second form of infection, extraintestinal form, in the liver abscess, characterized by remittent fever or sometimes high. Second, upper abdominal pain. Third, liver enlargement, elevation of diaphragm. Fourth, general weakness and other symptoms, large liver abscess that are not treated in time are often lethal. Third form of infection of extraintestinal amoebiasis are much rare and include the involvement of lung, brain, and the skin. Let's move to the lab diagnosis. Intestinal amoebiasis can be diagnosed by stool examination, microscopically, and stool culture. Stool culture is useful especially in the cases of chronic and asymptomatic intestinal infection, when the excreting less number of cysts in the feces. Or we can diagnose by biopsy specimen, should also be submitted for histopathological study. Can also diagnose by molecular methods, which means DNA probes and specific sequence can be amplified by polymerase chain reaction, PCR. This useful for differentiate the Entamoeba histolytica from other intestinal protozoa, including Entamoeba coli, Entamoeba dispara, Entamoeba hartmanni, or Entamoeba gingivalis, Entamoeba coli, Endolimax nana, and Idiomorphia pochita. Also, we can diagnose by blood examination. It shows moderate leukocytosis. Serological test. This test is negative in the early cases. However, in late lateral stage of invasion intestinal amoebiasis, antibody appeared and serological tests become positive. These include indirect hemagglutination test, indirect fluorescent antibody test, enzyme link immunoabsorption assay, ELISA. Hepatic amoebiasis can be diagnosed by aspiration, so trophozoid of Entamoeba histolytica may be demonstrated by microscopy of the pus. Aspiration by picture of amoebic liver abscess in less than 15% case, or by liver biopsy. Also, we can demonstrate the trophozoid. Blood examination It shows leukocyte with the total leukocyte count 15,000 to 30,000 per microliter, of which 70 to 75 are polymorphic nuclear leukocyte. Also, stool examination in less than 15% cases of amoebic hepatitis, cyst of Entamoeba Histolytica can be demonstrated in stool. This indicates the persistence of intestinal infection. Also, we can diagnose hepatic amoebiasis by serological test and histology. This test can be made when the trophozoid within the tissue also can diagnose by molecular methods such as DNA probes and PCR or polymerase chain reaction. Treatment. We can treat the Entamoeba Histolytica by very effective drug for the therapy of amoebiasis is metronidazole or its analogs tinidazole and or nidazole. Because metronidazole is so well absorbed in the gut, level may not be therapeutic in the colonic lumen, and the drug is less effective against cysts. Hence, patients with amoebic colitis also should receive luminal agents to eradicate the Entamoeba Histolytica trophozoid residing within the gut lumen. The non-absorbed aminoglycoside, polymycin, and 8-hydroxyguanidine compound, idaguanolone, are effective luminal agents. We can control on Entamoeba Histolytica or prevent it by improving the environmental and food sanitation. Treatment of carrier is controversial, although it is agreed that they should be barred from food handling. Personal and public cleaning. Not fully satisfactory and safe drug is yet available for chemoprophylaxis, and the list of drugs required for therapy attests to the problem of treating amoebiasis. Finally, the conclusion, to sum up, we're talking about Entamoeba Histolytica, which is pathogenic intestinal protozoa, belong to the Sarcodina, the amoebic pseudobody. It has two stages in its life cycle. These are trophozoid cysts. It causes 3 types of infection. These are asymptomatic infection, dysentery, and extraintestinal infections such as liver abscess. It can be treated with metronidazole or its analogs tinidazole and or nidazole, as well as aminoglycoside, polymycin, and the 8-hydroxyguanidine compound, idaguanolone. Thank you for your listening.